In recent years, antibiotic-resistant strains of gram-negative bacteria have been identified as urgent public health threats. Patients who receive delayed appropriate therapy are more likely to have an infection due to antibiotic-resistant pathogens, and patients with infections due to antibiotic-resistant pathogens have also been shown to experience poorer outcomes, compared with patients with infections due to antibiotic-susceptible pathogens. Introducing Zavicefta, a combination of ceftazidime, an anti-pseudomonal cephalosporin, and avibactam, a novel beta-lactamase inhibitor. Ceftazidime-avibactam provides broad-spectrum coverage against multiple resistant pathogens. Zavicefta has broad-spectrum coverage against gram-negative pathogens, including in vitro activity against multi-drug-resistant, MDR, Pseudomonas aeruginosa, carbapenem-resistant Enterobacteria ca, CRE, including KPC and OXA48-producing strains and extended-spectrum beta-lactamase, ESBL, and AMP-C-producing bacteria. It is indicated for infections in adults with complicated intra-abdominal infection, in combination with metronidazole. Complicated urinary tract infection, including pyelonephritis. And hospital-acquired pneumonia, including ventilator-associated pneumonia. Let's hear what the experts have to say about Zavicefta.
It's a difficult and treacherous enzyme because the levels of resistance that it gives to carbapenems are often very low. It can easily be missed by the microbiology laboratory. What's more, if an isolate has only got OXA48 and no ESBL, it'll still be susceptible to cephalosporins, particularly to ceftazidime. Several clinical studies have shown that OXA48 producing Enterobacteriaceae are associated with poor outcomes. A couple of studies, 30 to 40 patients each from Spain and in Turkey, 50% mortality in bacteremic patients with OXA48 producers, largely treated with mixtures of carbapenems, aminoglycosides, colistin. We'd need treatments for these infections. And that brings me to the topic of ceftazidime-avibactam. Adding avibactam really gives a new lease of life to ceftazidime, which was an old and highly regarded antibiotic. Avibactam overcomes ESBLs and AMP-C enzymes, which are the commonest modes of resistance to ceftazidime. Avibactam also overcomes KPC and OXA48 beta-lactamase.
Well, the importance of coverage, a very wide range of pathogens nowadays is something that it's absolutely critical. Because the epidemiology nowadays is quite complex in many institutions around the world, and there is a clear increase in multi-drug-resistant gram-negative, particularly causing pneumonia, causing urinary tract infections or intra-abdominal infections. And at the same point, it's very important to remember that the empirical antibiotic that we provide to our patients is very critical for the outcome of our patients, and of course, multi-drug resistance is decreasing the probability that the drug you are choosing, of course, it will be effective from the very beginning. Ceftazidime-avibactam is an active anti-pseudomonal agent with activity against 75% of Pseudomonas species isolated in Greece, Italy, and Spain, including those with multi-drug resistant and extremely drug resistant patterns. The current clinical trials do not capture the real-world cases that benefit from ceftazidime-avibactam, but demonstrate that this antibiotic is effective and well tolerated as any other beta-lactams for susceptible microorganisms.
Colistin and polymyxin B is an older antibiotic. We didn't use polymyxin for more than 50 years in medicine. But when CRE appeared, there was no other choice. So polymyxin became the cornerstone, became the backbone to treat CRE before the new antibiotics. But the problem is that polymyxin has significant limitations concerning the safety profile, particularly neurotoxicity and renal toxicity. And recently, many centers have published the increase of resistance. Avibacter is a novel beta-lactam inhibitor that has very good activity against KPC and many OXA48 isolates. It does not have activity against methyl beta-lactamase.
So for this reason, my personal idea is that when we look at the community-acquired infections, I think there is a role for tigecycline in low to moderate severity, but absolutely important, but also in high severity. And you can see here the role of tigecycline in both the situations. The only way is how to use tigecycline, and to use in monotherapy or to use in combination. So I believe that in community-acquired infections, we can use in monotherapy. In healthcare or in healthcare associated infection, again in monotherapy, probably where we have to use the combination therapy with another agent maybe active against Pseudomonas, we have to use in hospital-acquired infections. So the patient profile for tigecycline, in my opinion, is represented by elderly, particularly with comorbidities like diabetes, pen allergy, van intolerance, peripheral vascular disease, renal or hepatic impairment, on multiple medications. And then I think that particularly good for patients with prior antibiotic exposure, patients coming from nursing home resident, patients with a recent hospitalization, patients with a history of MDR. So in conclusion, is there a future for tigecycline? There was an editorial that I've written 6 years ago, and I think the increased medical need represented by the growing impact of multi-resistant infections and the current lack of alternative or new antibiotics suggests that tigecycline benefit risk continue to be positive. And in my opinion, we don't have to forget that tigecycline is a potential tool to save carbapenems, either as a primary treatment or de-escalation, and tigecycline should also be used to avoid collateral damages. So in summary, the value of tigecycline in intra-abdominal infections is that I think that there is a risk. Intra-abdominal infections are life-threatening infections with a rapid rate of progression. There is an importance to speed up therapeutic management. Tigecycline offers a broad spectrum of activity against pathogens causing abdominal infections. And in these real-life studies that I show you today, tigecycline alone and in combination achieve favorable clinical response rates in patients with abdominal infections with the high severity of illness. And tigecycline as a tool to save carbapenems is an important message, in my opinion.
Here at Pfizer, we have a long history of discovery, development, and distribution of anti-infectives, and we are committed to delivering the highest quality products for your patients. As the originators of piperacillin tazobactam, we are pleased to continue supply of Tazocin EF piperacillin tazobactam for your market after making important changes at the key manufacturing facility in Catania, Italy. For the past 170 years, we have helped to shape the pharmaceutical industry. For over 75 years, we have contributed to the treatment of infectious diseases, and we now have a wide portfolio of anti-infective products. The defining moment for us was in 1941, when we became the first company to invest in new production processes. To manufacture mass quantities of the first real defense against bacterial infection, penicillin, originally discovered by Alexander Fleming in 1928. We have an established history of high-quality production and proven effectiveness in imminent life-threatening bacterial disease. We have confidence in our high-quality products, and this high quality is managed via a comprehensive and robust quality management system. Not only are we committed to ensuring the highest quality, we are also dedicated to environmentally sustainable practices aimed at reducing our carbon footprint, increasing energy efficiency, decreasing dependence on limited resources, and reducing waste. We are proud to say that since 2000, we have reduced our greenhouse gas emissions by 60% and aim to reduce this even further by 2020. The manufacturing process of Taserson EF starts from the solution preparation phase. At this stage, active pharmaceutical ingredients are mixed along with excipients until the chemical reaction is complete. The resulting solution is filtered by a sterilizing filtration step. Following this, vials are filled with a sterile solution by a filling machine and pre-stoppered. The vials containing the liquid doses of Taserson EF are then loaded into freeze dryers to undergo a lyophilization process. After completion of the lyophilization cycle, the vials are moved into a capping station for the last manufacturing process step, the application of the external cap. The sealed vials are then passed through a 100% visual inspection process before the final packaging is performed. Taserson EF is indicated for the treatment of the following systemic and/or local bacterial infections in which susceptible organisms have been detected or are suspected: lower respiratory tract infection, urinary tract infections, complicated and uncomplicated, intra-abdominal infections, skin and skin structure infections, bacterial septicemia. Polymicrobic infections, including these where aerobic and anaerobic organisms are suspected, intra-abdominal skin and skin structure, lower respiratory tract. Taserson EF in combination with an aminoglycosides is indicated for bacterial infections in neutropenic adults or children. Currently, a non-EDTA formulation of Taserson EF exists. However, in 2005, Taserson EF was uniquely reformulated to include the addition of EDTA, a metal chelating agent. This formulation enables reduction in nurse administration time, reduces the need for a second IV line, as it can be co-administered with aminoglycosides, amikacin and gentamicin, via Y-site infusion, and decreases the use of disposables, therefore indirectly reducing hospital costs. Taserson EF is an effective and well-tolerated treatment option for the treatment of approved indications. We are delighted to be able to resume the high-quality production of Taserson EF and continue to provide healthcare professionals with this important product in our anti-infectives portfolio. In January 2016, we pledged our commitment to tackling antimicrobial resistance by joining with over 100 industry partners to sign the Declaration on Combating Antimicrobial Resistance, announced at Davos, to call on governments to join industry in taking swift, coordinated action. At the core of our efforts, we are actively involved in ongoing surveillance programs that monitor key pathogens and antimicrobial susceptibility patterns across different regions. These programs continuously monitor the in vitro activities of our anti-infectives portfolio around the world to provide susceptibility and resistance updates to the medical community. One such valuable resource is Atlas, Antimicrobial Testing Leadership and Surveillance, available online and via an app, which provides visual representations of key surveillance information and trends on bacterial resistances dating back to 2004.